thank you so very, very much. This has been an exciting symposium, and thanks to the leadership of Charles Lee and his colleagues here, this has not only been a very educational one for me, but it's after having been in Asia for over a decade, I've been remarkably impressed with the developments in Korea in terms of not only the type of experimentation, the scale, and the impact of the work that you're doing, exemplified by the last several talks that you've heard. I'm going to continue the theme of the discussions about systems biology, systems genomics, using breast cancer as a model system. I believe that the great challenges for this decade and the next will be to understand genomic complexity, not in terms of just understanding the components of that complexity, but how those mutations actually interact with each other. Secondly, how those interact in the, very much as what Dr. Lee has mentioned, how does it make a cancer cell more robust? Robust not only against the natural protections by immune system, but also robust in terms of any therapeutic challenges that we might throw at it. And part of that has to be an understanding of cancer evolution, which you're already hearing from Dr. Kim, how the heterogeneity may play into that. In order to do that, I think the tools that we need to develop is a better understanding, an operational understanding of systems genomics. What I mean by that is that we have an operational understanding of how to take a candidate gene, put it into a cell looking at the mutational effects, but how do you then experiment when you're dealing with 10, 20, 100 mutations simultaneously? How do you define the importance of a cassette of so many different components? Single cell biology will allow us to look at the evolutionary framework of a cancer and its how heterogeneity may play into that. And we have to have reliable model systems. And of course, we're very keen on the PDX model because it removes us from the artificiality of cancer cell lines that have been passage over many years. Now, the core to systems biology in experimental fashion is really to understand combinatorics. Combinatorics meaning combinations of 2, 3, 4 oncogenic elements and how it functions to generate a cancer phenotype. Now, the concept of combinatorics is really well known, established in cancer. For example, one of the first examples is that you need MYC, overexpression of MYC and a mutant RAS in order to transform rat embryo fibroblasts. That's been known a long time. Human cells are more resistant to transformation, require other mutations to take place. And this was done by the Weinberg lab, an SV40 large T, which knocks out P53 and RB, mutant RAS and HTERT to simultaneously be turned on in order to activate, to transform human epithelial cells. So my talk will be focused on one observation that we've made, that the chromosome, your chromosome, has an architecture that is evolutionarily determined, and that this architecture can be parlayed by a cancer to be an oncogenic organizer that coordinately alters gene expression of oncogenes and tumor suppressors. And I'll show you some of the details around that. In fact, I will focus primarily on a genomic configuration. Actually was mentioned by Dr. Lee, a segmental duplication, but in this case what we will call a tandem duplication of a fragment of DNA. It can encompass not a gene, but within a gene. It can encompass a single gene, encompass multiple genes. And in this framework, it's usually a single copy, head to tail copy of a segment of DNA. That's what I will call a tandem duplication. What we have found over the last few years that tandem duplications are probably the nucleating force of amplicons in cancer, that it generates very frequently fusion transcripts, and I'll tell you how, and we've found a tandem duplicator phenotype, which has been observed before, but actually have shown that this is a coordinated systems approach that a cancer uses to simultaneously mutate a number of oncogenes and tumor suppressors at the same time. The amplicon I will look at is HER2/neu, and you already know that's a receptor tyrosine kinase amplified in about 20% of breast cancers, associated with worse survival, and is clearly the target for effective antibody therapeutics, which was really one of the origins of precision therapeutics. Now, we started to look at this on a genomic scale, and this is just data that had come out of the Broad Institute on 243 breast cancers with HER2 positive configuration. And what you see here, red is increased copy number, blue is reduced copy number. And you can see that there's already, you don't need mathematics to look at this, but to see that there are regions that have consistently low copy number, consistently high copy number, low and high. And it turns out that these are in regions of known oncogenes important in breast cancer. For example, P53 is on 17P, that's frequently lost. HER2 is amplified here. In between this is an element of single copy deletion. That's where BRCA1 resides, and it's lost in copy. And then from 17q21.3 to the telomere is this another coamplicon. Now, as a geneticist, you would look at this and say these could be and should be viewed as independent events. But I'll show you that it's not, and it's organized by the chromosome itself. When you look at the conjoint events of gain of ERB2, loss of BRCA1, amplification of 17q21.3, and something that is lost is TP53, that the combination of all four cannot be by chance alone, is highly statistically significant. It's an odds ratio of almost 8. You don't see this in any other configuration in breast cancer. So we started to ask the question by using deep genome sequencing. At that time, we were using Yijun Ran's distant paired end sequencing the ends of 10 kb fragments, which allowed us at that time to be in a parsimonious and cost-effective manner to have deep sequencing for structural mutations. In this way, we could use standard statistics to ascertain significance. But what we found was that for HER2 amplified tumors, there frequently was amplification of HER2, but a deletion or an inversion deletion that encompassed the BRCA1 that joined this fragment, that 17q21.3, with ERB2, and this region was co-amplified. When we looked at it more carefully, in every single case of HER2 amplification, there was actually a tandem duplication that nucleated that amplification. By just simply counting, you can do genealogy of that amplicon, and that tandem duplication, that breakpoint, because we can map the breakpoint, was actually the centerpiece of that amplicon. So what you have in multiple examples is a tandem duplication of ERB2 followed by a deletion or inversion deletion that encompasses BRCA1 that joins this duplicated ERB2 with a segment 17q21.3, and that element is actually co-amplified. So this is how it would be schematically happen. P53 is somehow lost. That's seen very frequently. BRCA1 is lost. These two are joined, and its amplification takes place. Now, the postdoc who was working on this project, a brilliant gentleman by the name of Koichiro Naki, who's now back in Tokyo, was actually an origami enthusiast. And he called this chromosomal origami. So if you wanted to cut 6 holes at once, you can do this independently, or you can fold the paper and cut once and open it up, and you'll have 6 or more holes. So we call this chromosomal origami to generate cancer gene cassettes. Now this suggests that there are, in this 17q1.3 to the telomere, cancers don't do these kind of amplifications, deletions simply on a random basis. These things either have a neutral effect on cancer growth or has a positive effect, even though the positive effect may be relatively small. So we went and redefined what we would call oncogenes and tumor suppressors. This is work done almost a decade ago in the Genome Institute of Singapore with my colleagues, where we took a data set of array data set, expression array data set of over 800 breast cancers with clinical annotation and correlated the expression of each gene with survival of the individual. If the gene is expressed beyond median, overexpressed, and it's associated with worse survival, we called it an oncogene, just definition. If it's overexpressed and it's associated with better survival, we called it a tumor suppressor gene and reannotated the human genome based on that correlation with clinical outcome. And what you see here is that in an amplification on chromosome 8, which culminated in the MYC, we had when each gene were looked at, there was a predominance of poor prognosis, which is oncogenes, and in 8P, there was a predominance of good prognosis genes. So this is one way to annotate this. When we looked at 17, of course, a very sharp peak occurred at ERB2, reconfirming what we've known for a long time, that ERB2 is a major driver. We noticed that there is in BRCA1 a signal for a tumor suppressor, but this suggested that all along the 17q21.3 to telomeric amplicon had a number of proto-oncogenes that might support cancer growth. Now, what Cochero did was heroically took a whole number of breast cancer cell lines and did siRNA on them, and simply accumulated all the results of the effects of this siRNA on growth. So if it's an oncogene, siRNA would reduce growth. If it's an anti-oncogene, siRNA would increase growth. And what you found here is that, gratifyingly, ERB2 was the most oncogenic element, using growth as the measure of oncogenicity. But that 19 out of the 21 genes on 17q21.3 to the telomere had oncogenic potential, with only few with anti-oncogenic potential, suggesting that that co-amplification was the amplification of oncogenic elements in a simultaneous manner. What about BRCA1? I told you that seemed to be coordinately lost when ERB2 is amplified. So we use human mammary epithelial cells, or HMEC cells, that have been either knocked down with BRCA1 or have HER2 transfected into it and then had BRCA1 knocked down. What you see here is that in shRNA, control shRNA, this would be the baseline. When shRNA for BRCA1 was given to those cells, it increased the growth of those cells. This is HER2 growth when HER2 was transfected into those cells. When HER2 plus shRNA was introduced into the cell, you had even further growth, suggesting that the conjoint amplification of HER2/neu with the suppression of BRCA1 had at least additive effect in growth in these human mammary epithelial cells. Now, this suggested also that there might be some effect of these other ancillary genes on the effectiveness of anti-HER2 therapeutics. Now, antibody therapeutics are not well experimented with in these cell line experiments. So we used a drug, lapatinib, that was targeting HER2 signaling as well as EGFR signaling. HER2, by the way, is a kinase negative gene, but it works by combinatorics and dimerization with other members of that family. We simply asked the question, is the effect additive to lapatinib, which would lie on the x-axis? Is it synergistic, which will be above the x-axis, or if it's antagonistic to lapatinib, if it's below the x-axis? What you see here is when HER2, which is a target for lapatinib, is knocked down, you would expect it to be antagonistic because the target is gone. All right? But when we looked at it, the majority of the 19 genes that we tested here were either additive or synergistic with lapatinib, suggesting that these genes that are co-expressed or co-amplified with HER2 has an effect on the effectiveness of the HER2 therapeutics. When done on a control control genes in a chromosome that is not affected in breast cancer, chromosome 2p, there wasn't any significant changes in terms of additiveness or antagonism. Now, this actually raised a very interesting conceptual point, and that is for those of us who were raised in the oncogene era, when we had fundamentally only a few oncogenes that really worked, we always thought these as driver oncogenes, HER2, and everything else in the amplicon as simply a passenger mutation. But what I'm showing you here in an evolutionary context, that there are indeed very important driver oncogenes, but there are many backseat driver oncogenes that have much less effect. But if you think about it as an evolutionary biologist, it doesn't take much to, a 1% change in robustness will over 100 generations enrich for individuals with that one variant that gives you only a 1% change. Okay? And so consequently, what we're looking at is an optimization by the cancer cell to fight off the challenges that they face with the natural immune system, with the various other control mechanisms to control cancer growth. And it requires the conjoint interaction of many oncogenes and tumor suppressor genes to have that happen. In this case, however, these are not independent events, but events that have been organized by how these genes are juxtaposed with each other within your genome. Now, what about the concept of gene fusions? Now, it turns out that when we were doing some RNA-seq simultaneous with this distant paired end sequencing, we found that there was a large number in these breast cancer samples of what we call out-of-order fusions, very much like what Dr. Lee had described in CTLA4 and CD28, where gene A and B, if there was a splice abnormality, the RNA would still have the order A 5', B 3'. But we saw a number of these fusions where the B is 5' and the A gene is 3'. And there's no way this could occur except by this kind of tandem duplication. We were able to go back and validate that by virtue of the genome sequencing because there was a breakpoint that was there. We were surprised to find out that 65% of the fusions tested that we found in a number of cancer cell lines came from these tandem duplicons. Now, they may be expressed at lower orders or even at higher levels, but they existed. One thing that one fusion transcript we were very interested in was this one also on 17q. It was TMEM and S6 kinase, where it looked like there was a tandem duplication, a break, as you see here, and a fusion transcript that had the S6 kinase fused with this adjacent TMEM transmembrane protein. Okay. When we saw this, we said, wow, could this be another BCR-ABL rearrangement or EML-ALK? But what we found was that was not a case at all. There was heterogeneous mutations that we saw. And in particular, in this case, the kinase domain was not included in the fusion, and the transmembrane domain was not included in TMEM. So it didn't make a whole lot of sense. But one thing that was very intriguing, we've known for a fact that the amplicon and in 17q that encompassed S6 kinase was associated with worse survival in breast cancer. And later on, and when we looked at the presence of this fusion, this out-of-order fusion, which included different exons in different tumors, but yet the same order was affected, it was found in about 30% of of breast cancers that we tested, and these were also associated with worse survival. Now, what was this all about? None of these made sense in terms of the fusion structure, and yet it was associated with worse survival. We took the cDNA of this out-of-order fusion, several of them, transfected it into cells, and found no change in the tumor biology. And so what was really going on? Well, we looked at around left and right of the genes around TMAM and S6 kinase. And what we found here that in all cases, now these are in primary tumors, when we had quantitative PCR, that the adjacent gene, which is microRNA 21 and S6 kinase, whenever there was a fusion, there was higher levels of expression. Now you can say, well, they were broken, the S6 kinase was broken into, but there was always an intact S6 kinase in that tandem duplicon. And for whatever reason, perhaps more open chromatin or otherwise, there was higher levels. But we also found out that whenever there was a break, there was also an amplification. Recall, the tandem duplicons are nucleating amplifications, and this was true here as well. Why microRNA 21? Well, remember, TMEM is right next to it, but what I didn't tell you was the microRNA 21 was tucked immediately 3' to the TMEM gene. So whenever there was this tandem duplication, TMEM itself was involved in this rearrangement, and there was overexpression. So what you have here is this tandem duplication, and in fact, we call this fusion a sentinel fusion, a signal fusion, not a driver fusion, but it signaled that this region was ripe for a tandem duplication and amplification. And the amplification was not only on S6 kinase, but a known oncogenic microRNA, which is microRNA 21 as well. Now, an important part of these fusions, you can say, well, okay, then they're really not important. But we did a further experiment on MCF7 cell lines and asked, what is about these fusions? Okay, MCF7 had a number of these tandem duplicons, and many of them resulted in a fusion transcript. So we did polysomal fractionation. Remember, polysomes are for translation. They lock onto RNA that it might be translated, and it's the high polysome fraction as opposed to the monosome fraction that is associated with translation of RNA species. And it's been shown that by centrifugation you can isolate the polysomes and do quantitative PCR of the RNA species that are locked onto this polysome. And what we found was that 50% of the fusion transcripts that were in MCF7 are in the in the polysome fraction, which suggested that they would be translated. Now, this is actually very interesting. By the way, it wasn't just this fusion transcript, it was all the fusions that were found in MCF7. Why is this kind of important? For the immunologist, you know that these fusion transcripts are candidates for neoantigens, right? Okay. And this is actually one of the very interesting observations. You heard about the humanized NSG mouse, where the human immune system was placed into an immune-deficient mouse. Subsequently, 12 weeks later, a human tumor is placed into that. And when we looked at this in a PDX tumor, we found that in this one case, recall that breast cancers are not known to be terribly responsive to anti-checkpoint inhibitors, but with anti-PD-1 and anti-PD-1 plus CTLA-4, we found a significant response, as you see here in this humanized mouse. When cisplatin is given in this case, which is a triple-negative breast cancer, it was always also a response. Now, the question is, why should this be the case? Recall that now there's a growing hypothesis that the reason why some tumors respond and others is because of mutational load. That the higher the mutational load, the more neoantigens that might be available for recognition in the immune system. And we're suggesting that these fusion transcripts of which at least 50% are going to be translated, may be a source of these neoantigens, and this being tested in conjunction with the Carolina Palooka that Charles talked about this morning. Now, this raised yet another framework of tandem duplications, and that is the concept of chromotypes in cancer. What do I mean by chromotype? Now, you heard about, let's say, gene amplification or single tandem duplication, but you have these unique chromosomal configurations where entire chromosomes are jumbled up simultaneously and not in a successive fashion. And the first one that was described was chromothripsis, where an entire arm of the chromosome is literally fractured in incomprehensible ways and reconstructed. And yet, upon reconstruction, by the way, MYC on chromosome 8q is a target of this chromothriptic reconstruction. Well, we found that there is others have found earlier that there's a tandem duplicator phenotype whereby the whole chromosome is dotted with many single tandem duplications, not amplifications, but single tandem duplications across the genome. We actually developed a quantitative measure of how to identify tandem duplication phenotype from just all other tandem duplicons, because there seems to be a bunching up of those tumors with this kind of phenotype, and not just having tandem duplications that are amplified in one location. We find that they're enriched in triple-negative breast cancers, in ovarian cancer, and in uterine cancer, and depleted in many others, including prostate, glioblastomas, and lung cancer. This meant that there must be something about the tandem duplicator that seems to be linked with these cancers. We then asked, is there some kind of systems strategy for this, or are these tandem duplicons just random within the genome? What we found using multiple sources, not only sequence sources, but array sources, now over in examining over 500 tumors, that the genes within the tandem duplicons tend to enrich for known oncogenes, Go genes that have been described before, or putative oncogenes by other methodologies. Whereas in the boundaries, now recall when you have amplifications, the boundaries are subject to mutations and deletions. The boundaries are enriched for known tumor suppressor genes that's there. So a single tandem duplication would then coordinately amplify a oncogene and delete or repress tumor suppressor genes as well. This also is associated with expression. Those in the center of a duplicon tends to be more expressed than the baseline, and those at the edges of the duplicon tends to be repressed from the baseline, consistent with the oncogene and tumor suppressor gene configuration that I told you about. I'll just give you a few examples of this. One is MYC, which is actually an oncogene. We know that this is a relatively common area of duplication, and you see here from about 400 of these tumors, this is the range or the span of the duplicon that is seen there. And you can see that MYC is directly in the middle of every single one of those duplicons. In P10, very interestingly, each of these duplications interrupted P10. So it's a beautiful way that the genome has allowed for knocking out of tumor suppressors and overexpressing oncogenes in a conjoint manner. So to end, I'd like to acknowledge the important people. Koichiro Inaki, who is now back in Tokyo. Francesca Mengi, who joined me from Singapore, is fantastic, both biologist and computational biologist. Pooja Kumar, who did a lot of the cell culture work. Fung Schreckengrass is our technical assistant. Jeff Trong did a lot of the computational work, and Ijim Ran, the technologies. And the PDX that you heard about is Jim Keck and Carol Bolt. So I want to thank you very much. Congratulations on a wonderful seminar.